Good evening and welcome to tonight's lecture on sample size and power. My name is Dr. Laura Lee Johnson. I'm an Associate Director at the United States Food and Drug Administration. Oops. Who apparently can't remember which clicker she's supposed to use after 15 years. So the disclaimer, of course, nothing that I say the FDA wants to be construed as representing their views or policies. So why do we care about sample size and power? Let's think about this a little bit. Now, last week you all heard a couple of lectures from Paul Joachim. This is actually a slide that he showed in another set of lectures that we gave. And I think it's a very nice way to kind of set the mood for this lecture. If you forget by the time we go through all the formulas in the next 90 minutes, power is basically the probability of getting a statistically significant result when in fact there's a clinically meaningful difference. So this is kind of unknown to us, right? And part of what we're going to be discussing over the next several weeks is how do you know that something's clinically meaningful? But this, in essence, this idea of power, we want to have high power, right? We want to have a high probability of seeing a statistically significant result in those hypotheses tests from last week if, in fact, there is this clinically meaningful difference. So by definition, those studies that have low power are less likely to produce statistically significant results, even when that clinically meaningful effect exists. And that's the reason your animal care committees and the human subjects committees are going to say, well, is it ethical to do research if you don't have that much power? So this becomes not just a, I want to have significant findings question, it's also an ethical question. Is it worthwhile to put people at risk or their data or their privacy even at risk if you're not going to be able to tell that, as one of my colleagues said, there's a there there? Now, the flip of this and how power and sample size and statistical significance tie together is that the lack of statistical significance doesn't prove that there's no treatment effect, because it could be a consequence of small sample size, or really, it's low power. So it's important to have enough power and an adequate sample size. So our objectives for this lecture are to calculate changes in sample size based on the changes in the difference of interest and the variance or the number of study arms. So we'll talk about each of those. We'll understand the intuition behind power calculations, hopefully, by the end of tonight's lecture. Be able to recognize some of the common sample size formulas for the tests that were talked about last week, and learn some tips for getting through the IRB. Your general takeaway message is you need to get input from a statistician. It's not just because I am one, but because the ramifications of not continually talking to your statisticians and epidemiologists can be pretty big. You can waste a lot of money and a lot of time. All calculations, after I say calculations are important, you kind of have to take them with a few grains of salt. Most of the basic formulas that you will find are like the lowest level possible. But in reality, we have these kind of what we call in the US fudge factors. So realistically, people are going to drop out. Maybe the variance of that endpoint is going to be larger than you were anticipating. Maybe accrual will be slower than you were anticipating. A lot of things can happen. And so we have to take that into account when we're trying to figure out what our actual sample size should be. The other trick is you will always, always, always round up on sample size, never down. Why is that? Because if your calculation tells you that you need 10.01 human beings in your study, there is no 1/100th of a human being. You need 11 people in your study, or your power goes down. We'll talk a little bit more about that as we progress through the lecture. But also always remember that analysis follows the design. So a few other take-home messages. You really need to give thought and have a broad discussion about what difference is scientifically important and in what units Are we talking inches or centimeters? Why? It can actually, these sample size calculations are not invariant. It's a mathematical term, but the units matter. And also the measurement error. Remember I mentioned at one point, do I use like a little plastic ruler, or how other method could I use to measure my waist? Think about how you're going to be measuring, because that information has to go into your sample size calculation. Are you taking a single measure of systolic blood pressure? Are you averaging 3 together? This information is going to matter when you're calculating power and sample size. How variable are the measurements? Again, my favorite plastic ruler does not measure my waist well. So in sample size, you need to think of a few elements. One is, what is the difference or the effect to be detected? This might be a ratio, it might be an actual difference, but this element needs to be known. You need to think about the variance in the outcome. You need to understand what significance level you want. So is it a one-tailed or a two-tailed test is also an important part of all of these equations. What power do you want? What level of type 2 error are you willing to allow? Do you want equal randomization to different study arms or unequal randomization to all the study arms? Are you thinking about doing a superiority, equivalence, or non-inferiority trial? All of these have different sample size calculations or different numbers that you need to put in for the calculations. Other elements that come up will be a follow-up period. How long a participant is followed. One of our examples will talk a little bit about that. We'll also talk a little bit about censoring. This is when a participant has been in the trial for a while, you have all their data, but now that participant is no longer being followed. So you have what's called incomplete follow-up, which is common, and sometimes we also do administrative censoring. So let's say I'm trying to follow people and find out when they die. Well, it's the end of my study and they're still alive. So at the end of study, I may administratively censor them, saying I knew they were alive up until this time point. We'll talk more about this in the survival analysis lecture. And here are a few other questions from Paul. For those of you in the room, you can just yell this out, but I'll say it orally too. Without what is there no need for statistics? That's right. Without variability, or variance, there's no need for statistics. Actually, a friend of mine at NIST, the National Institute on Standards and Technology, told me this. He's like, my job is to make everything so error-free that statisticians aren't needed. And this is part of this issue with variability, right? You've got the old treatment and the new treatment. What we tend to do is compare their averages. But it matters if those dots, if those samples we have are really clustered tightly, they overlap just a little bit, they overlap a lot. These are different. It's harder to tell this apart than that first example. And also for those of you all developing those diagnostic tests, and you say, but, my sample size and my odds ratios, they look so good. It's like, well, what you really need to do, though, is distinguish new and old. And can you do that? So we're going to start off tonight talking about power, go through some basic sample size information, a whole bunch of examples, but there are going to be more examples in the textbook. I'm going to talk about some of the few changes you can make to the basic formula that's also covered more in depth in the textbook. You also will do a little bit of multiple comparisons. You've already talked about that some, and other people will talk about it more. So I'll try to race through that to get to what St. George's College has put out in their information to researchers as these are really poor sample size statements. Please never send them to our IRB again. So again, power depends on your sample size. A lot of people ask me why we don't have power formulas. Well, they are there, but they're algebraically intensive. It's actually easier to teach the sample size formula. You can work backwards to figure out a power formula if you need it. But either way, the math will work. But again, this idea about power is it's the probability of rejecting the null hypothesis the alternative hypothesis is true. Typically, the more subjects you have, the higher power you have. So what's power affected by? I already mentioned the sample size, but variation in that outcome is huge. If I can lower my variance, I increase my power, all other things being equal. If I increase the significance level, so if I say, oh, instead of a type 1 error of 0.05, I could have 0.1, my power will go up. If I'm interested in different effects, it's always easier to detect a bigger effect. If I increase the difference I'm willing to detect, my power will go up. But again, it has to be reasonable. If you tell me that you're going to lower someone's average systolic blood pressure by 50 points, by 50 units, I'm going to say you are crazy, and that can't be done. So you have to be careful that your difference is actually reasonable. One-tailed versus two-tailed tests. For the same alpha, your power is greater in a one-tailed test than a comparable two-tailed test. Because remember, in the two-tailed test, I have to split it onto two different sides. So here are a few examples. Let's say that I have a two-arm study. I have a total sample size of 32, so I have 16 people in each study arm. So this is a small little study. Some people may think it's like an R21 or an R03 if you get NIH extramural funding. 81% power, my delta is equal to 2, so the difference I want to detect is 2. I set my standard deviation at 2. The type 1 error I set at 0.05, and I want to do a two-sided test. So at this sample size with all this information, I have 81% power. Let's say I was wrong on the standard deviation. Instead, it was 1, not 2. The good news is my power has gone up to 99.9%. Problem is, normally we are in the second situation where we thought it was going to be 2. Turns out it was 3. 2 and 3 sound so close together, but not in power land. Now my power's dropped to 47%. We'll talk about why in a few minutes. The significance level. I was going to plan my study for 0.05. Now they're saying, no, you have all these things going on. We think you should test at 0.01. Your power, instead of being 81%, is 69% when you do that. You cannot change your alpha level without changing your sample size if you want to maintain your power. But let's say instead you are going to power all of these interactions at 0.05, and somebody said no. You can test these interactions at 0.1, not 0.05. Well, your power's now gone up 81% to 94%. That's pretty cool. Let's say the difference to be detected. You wanted to detect a difference of 2 hours of sleep with your new intervention. You have this behavioral intervention, you think people will sleep longer. You cut down to 1 hour of sleep. Your power to detect 1 hour of sleep is 29%. Not such a smart thing. You kind of want to figure out, do you care about 2 hours or 1 hour? Let's say instead of 2 hours, you go for 3 hours. These people are getting no sleep, and you just want to see, does it show anything? Well, your power's gone up to 99%. But does your intervention really cause people to sleep 3 more hours a night? Probably not. Let's say, like I admit I did once, you thought the sample size of 32 was per arm and not total for your study, so you actually had 64 people in the study. The good news is your power's 98%. The bad news is they just spent a lot of extra money they didn't need to spend. Problem, however, is we always find ourselves in this other situation. Sample size is supposed to be 32. We're running out of money. We're having trouble recruiting. And we end up with only 28 people in the study. So our power that was 81%, and typically the reason I chose 81% is because 80% is usually considered kind of a minimum for a study, minimum level of power, although that varies. We'll talk a little bit about that. But what happens here is my power is now 75%. So I don't have 80% of a chance to see statistical significance when I have something that's clinically meaningful. I only have a 75% chance. Maybe that's okay, but maybe not. Then we have these two-tailed versus one-tailed tests. If I switch to a one-tailed test, I'll boost up a little bit to 88% power. So that gets us to what should your power actually be. So again, that phase 3 minimum tends to be 80%. There are other people that will say your type 1 error should equal your type 2 error. A lot of the large, huge, definitive trials might have 99.9% power. I have other folks that say, well, I'm just doing a proof of concept, and they have 60 to 80% power. And some of the omics studies that I look at, they have really high power because type 2 error is such a problem for them. So what is your power formula? All depends on your study design. It's just algebraically intensive, and you should probably use a computer program to figure out your power. Or make a statistician run a bunch of simulations for you to figure out your range of power. So this is kind of our basic sample size 101, where everything starts. Flipping from power to sample size, same elements are in play. Changes in the difference of interest are going to have huge impacts on your sample size, and so are changes in the variance. Some examples here, if you have a 20-point difference that you designed for, maybe you need 25 patients per group. If I change nothing else but this point difference, if I drop it by half to 10, I now need 100 patients per group. If it drops again to a 5-point difference, I now need 400 patients per group. All the types of things that feed into just your basic study design, a basic randomized 2-arm study. Changes in the difference to be detected. Your alpha, or that type 1 error, your beta, or 1 minus beta, your power. The standard deviation, the number of samples you're taking. Is it cluster randomized or non-cluster randomized studies? Is it a one-sided or two-sided test? All of these can have huge impacts on your sample size calculation. So your basic formula for the total sample of a 2-arm study, that's just simple, basic, individual, randomized study, nothing special. That 2n, where n is the sample size for a single arm, be 4 times, this is this critical value z. These are on that bell curve. Your computer will tell you what this value is, or if you have a really old stats textbook, there's a picture of the bell curve in the back that tells you. 1 minus alpha over 2. Why is this? Alpha, this is your type 1 error that you're going to allow, divided by 2 if it's a two-sided test. So for a 0.05 test that's two-sided, this value is like 1.96. Z of 1 minus beta, this 1 minus beta is that power. All right, so if it's 80% power, I think that's 1.645. Again, it's in your textbook. We actually wrote some of the numbers down. You take these two Z critical values, you square them, you multiply it by the variance, divide by the difference you're interested in squared. Now, all the different formulas for sample size, for all the different study designs and regressions, etc., they all look different, but these basic elements will be in every single one of them. So what do you need to think about before taking all this information to your statistician? What do you even need to talk to them about in addition to the basic background for your projects? First off, do you have a randomized or non-randomized study? Non-randomized studies require a lot larger sample size because remember, we've got all those potential confounders we've got to look at. Similarly, the effect modifiers. We have to account for that in the sample size planning. occasionally in surveys, so Barbara Stussman may talk a little bit more about this in a few weeks, absolute sample size may be of interest. So if you're doing a survey, sometimes we take a percent of population approach. We may say you have a given threshold and your confidence interval around your percent needs to be above a certain threshold. So you're trying to plan around that. So you've got to keep in mind, what is the study's primary outcome? That's your basis for your sample size calculation. That said, secondary or other outcome variables, if you think they're important, make sure you have sufficient sample size to be able to answer those key questions. Increase the real sample size for your study to reflect things like loss to follow-up, expected response rates, lack of compliance, etc. You can't do this, just off the cuff, though. You have to write into your human subjects applications and your grant applications how you are justifying those kind of plus-ups, right? And again, always round up. So let's say that you're going to do your sample size calculation and you're looking through books and you're looking online. What you're going to probably find are sample size calculations for two groups, a continuous outcome, looking at the mean difference. We're going to talk about that mainly tonight because the similar ideas hold for different types of outcomes and study designs, but do understand a lot of those formulas will not be applicable to your research because you're answering a different question. So to your lovely person helping you with your sample size calculation, you need to tell them what are all the variables of interest that you want to measure. What's the type of data? Is it continuous? Is it categorical? Is it ordinal? Bring them some examples if you can. What is the desired power? Where are you in this continuum of research? And what is the power that's going to be expected in your field? What is the desired significance level? Along those same lines, what's the effect or difference that's clinically important? Very rarely is this a single number. A lot of times what you're looking for is you're going to say, okay, what seems to be feasible given the research that's done? What seem to be consensus reports? Maybe you have animal data, you're trying to extrapolate it to human beings. Maybe you have nothing at all, and we'll talk about those. But what seems to matter? This is the reason I don't always like the effect size elements, where they just look at the ratio of the variance and the difference, or rather the standard deviation and the difference. The problem when you use that for your effect size is you haven't figured out, what's the actual variance? And does that effect size actually translate to a difference that's relevant? So think about those standard deviations for your continuous outcomes, but if at all possible, try to have real data on both of those bullets and decide if you're doing a one- or two-sided test. Realistically, you're going to have to have really good justification to do a one-sided test. What is your data structure? Are we talking paired data? Are you doing pre-post on folks? Are you taking repeated measures? Are you going to take this blood pressure measurement 16 times over a 2-year period? Are you looking at groups of equal sizes, or are you going to do a 2-to-1 randomization? Or maybe it's a case control study and you're going to get 5 controls for every case. Do you have hierarchical or nested data? Is that inside individually randomized folks, or are you doing a cluster randomized trial where you're going to have people inside of schools, inside of regions? Are you looking at biomarkers? Are you trying to actually do and develop a biomarker? Because there's a lot of stuff that can be involved in the laboratory assay part, and very different equations if you're in fact trying to validate a biomarker versus using it as essentially the endpoint in your trial. And how much validation information do you have about any of these endpoints? How do you know how they perform in your population? Your study design is going to really matter. I've talked a little bit about the randomization aspects. You need to talk to them about how you're going to randomize your trial. But again, they're all different formulations, whether it's equivalence. So again, how then do you define the equivalence bound? If it's non-inferiority, what's that non-inferiority bound? Or is it superiority? If you have a non-randomized intervention study, what other variables are you measuring that will need to go into the model? Is it prevalence or observational studies? Are you trying to figure out positive and negative predictive values, sensitivity and specificity? All of these have different calculations. So, a few of our examples. The short hint, because I always get asked this question, how many humans do I need when I know nothing? Sometimes the basic element has to do more with stability of the estimating equation than with really solving the problem. So you may run one of these calculations that tells you only need 3 people per arm. You will need more than 3 people per arm. 15 people per arm or rats per arm, etc., tend to be the minimum number. This is a good rule of thumb for early studies. Why is this? Well, when you have 12 to 15 values, you actually get a somewhat stable variance equation. I'm not saying it's right. It could be that you have weird sampling and the variance is off because of bias, but the equation itself for variance stabilizes between sometimes as low as 10, but usually 12 to 15 numbers in it. If you're using a Bayesian analysis technique, sometimes you need at least 70 people per arm before the analysis is in fact stable. A lot of our studies, a lot of our analyses and regressions float to something called a z-test. However, if you have a smaller sample size below 20 or 30, even sometimes below 100, you need to check a t-distribution. If you're doing something like logistic regression, principal components analysis, etc., you usually need a minimum of 10 participants per variable. These are individual participants per variable. And in some cases, you may need 100 per variable in order to get stable estimates. This is for the math will spew out of the computer. Doesn't mean the functions are stable. So there's this balance of wanting to find significant results. What do I need if I think these are the differences I'm aiming for? And then also, how many people do I need for the math to be sound? So now we're going to do, it's not truly a live statistical consult because I took screenshots, but we're going to look at sample size and power calculations. And actually, this isn't from this year's hypothesis testing lecture now, because Paul gave it for you all last week instead. But I'm going to talk about an example of cholesterol in hypertensive men. Let's say you have data on 25 hypertensive men. Mean is 220 for their total cholesterol, and you have a sample standard deviation of 38.6. If I go out and I look in national population data, 20 to 74-year-old male population, the mean serum cholesterol is 211 with a standard deviation of 46. So their standard deviation of the population is actually much higher than in my little sample. Not common, but that happens. The question is, my hypertensive men, is their cholesterol different from the average population? Now, it could be, because sometimes as we're treating hypertension, we're treating a lot of other things, including hypercholesteremia. So we're going to calculate the power with the numbers that are given. So what's the power to see a 9-point difference in mean cholesterol with 25 people? This is a question, is this a single sample or a 2-sample test? If I'm comparing to kind of population level information, this is really a 1-sample test. So Russ Length has sent up a whole bunch of different software. One's called PyFace. There are lots of different software programs you can use for this, but this is free, and it has a cute cow picture, so how can I resist? He also has some really great FAQs, or frequently asked questions, and other elements on his website. He also has a view that I deeply agree with, that please don't ask me for post hoc power. So he has a lovely article on why you shouldn't do post hoc power. Anyway, you've got this drop down for what type of analysis you want to do. So let's say you had a 2-sample test. Let's say I actually had 25 people that were normal that I pulled out medical records on, 25 people that were hypertensive that I pulled medical records on. I can set up all this information and figure out what my power is. So I can either set my sample size and calculate the power, or I can set my power and calculate my sample size. Well, let's do it for the 1-sample test. If I put in my sigma, I put in the true difference of means, if I went 90% power, I need 170 people. I basically have no power with my little sample of 25 to test for any types of difference in cholesterol. But you know, I kind of chose the 9 as a difference because that's what my difference was. But what is a clinically meaningful difference in total cholesterol? What should I be testing as that difference? These are all questions we would ask each other in advance of a study and not afterwards when you come to me and say, I have 25 medical records. This is the data I have. So what's nice about trying to design the study is I can move all these values around. Okay, you can't get 170 medical records. What can you do? What other assumptions can we make? So you can kind of see if it's not tenable at all to do this study or what your trade-offs are. So different study, going back to the 2-sample, which I meant to put that slide down here. Let's say I have a standard deviation of 2 in each arm, and you may not. Sometimes you have different standard deviations for each study arm. I'm going to run a 2-sided alpha 0.05 test. I'm going to look at a true difference in means of 1, 90% power, solve for my sample size. So sigma is 2, I say have equal. I'm going to 2-tailed test on equivalence. I define my alpha. Difference in means is 1. And if I want 90% power and I hit solve for a sample size, it's going to tell me I need 85 in each arm. But you can also make these nice little graphs. So you can say along the y-axis, this tall vertical axis, that's my power. And then I can see for the sample size in a single study arm on my x-axis how much power I have depending on where I am in the curve. So if I only recruit 50 people, what's my power going to be? These are nice curves to put in your IRB applications and your grant applications. So especially if you're asking for money, then you can say, listen, if you give me less money and I have fewer people in my study, this is the trade-off that's happening. Are you sure you want me to make that trade-off? So let's talk about dose escalation a little bit. Those, again, remember, we have basic sample size formulas. Most of us live in phase 1, phase 2. We're not going to use those basic sample size formulas as often. One of the simple phase 1 types of studies that we do are called dose escalation studies. We're looking many times in these first-in-human studies with a new compound for something called a dose-limiting toxicity, or a DLT. Now, it might be that this drug or this compound has been used in humans before, but now I'm trying it in a different patient group. A lot of these study designs came out of oncology, so they came from the cancer world. We've been modifying them over the years for kind of, less severe illnesses. And there are a lot of different ways to do phase 1 and phase 2 studies. But I'm going to go through what's called sometimes the 3x3, or there are a lot of other names, Fibonacci sequence, although it's not a real Fibonacci sequence. But these were kind of the basic models used for several decades for phase 1 studies, and probably a large portion of studies still today use them. but these have been modified in the last couple of decades to a little more sophisticated studies. But I want you to understand kind of like the basement where we all started. So in phase 1, you decide on a handful of dose levels. You decide kind of what toxicities are so bad you would stop and you wouldn't give people that dose anymore. So it might be that people die. It might be that, they have uncontrolled vomiting, whatever you want to call it. Usually you use something like the common toxicity criteria. Their AE grading, you say like anything that's a grade 3 or grade 4 AE, something like that. If you have a disease that's not that bad, you are going to be less willing to tolerate adverse events. And then we have some interventions that never cause toxicities, so you've got to figure out something else that you want to measure. Typically what happens in these phase 3, 3x3 designs is at least 3 patients will be treated on each dose level in each cohort. That's where this 3 comes from. This isn't a power or sample size calculation issue. This is, when it comes down to it, how many people are you willing to have on a totally unknown substance and basically kill at any given point in time? That sounds very ugly, and I've said it before, but I think it's really important to understand the gravity of some of the work that we're doing. And sometimes when there's no choice, you put more people on, and you are taking a higher risk. They need to understand those risks, and we'll talk about that more in the next segment of lectures. But an important element here is sometimes, even though we want to try 3 people in a given dose, we do it 1 by 1. Just in case 2 people die and they weren't supposed to, there's no reason to try it on a 3rd person. So kind of the old way that we would do this is we would enroll 3 patients. If 0 out of 3 patients have these dose-limiting toxicities, we'd escalate to the new dose. If you saw a dose-limiting toxicity in one of the 3 patients, we expanded the cohort to 6. So we added 3 more people to it because maybe it was a fluke. It may not be related to this drug that we're giving them. And we would escalate to a new dose if 0 of the next 3 new patients didn't develop that DLT. In other words, only 1 out of 3 at a given, or 1 out of 6 at a given dose developed a dose-limiting toxicity. The idea is that you are trying to work your way to what's called a maximum tolerated dose, or MTD. This is the dose level immediately below the level at which 2 or more patients in a cohort of 3 or 6 experience that dose-limiting toxicity. We're trying to go for a safe dose. Typically, the maximum dosage is pre-specified in the protocol. So we're going to stop at the MTD, or the maximum dosage. This is the big picture. We're going to go through it in steps. So first I enroll 3 people. One of 2 to 3, or 3 to 4 things can happen. Maybe none of them have a dose-limiting toxicity. One of them, or 2 or 3 of them have a dose-limiting toxicity. Of course, I drink water and my voice gets worse. So, nobody has a DLT. I escalate to the new dose. 1 out of 3 have a DLT. I enroll 3 more people at the same dose. And if none of those new 3 people have it, I escalate. If 1 or more in this new group have the DLT, depending on my risk tolerance, I may stop. But let's say I only had 3 people on that lower dose. I don't really know that much. So what I might do is, in fact, drop down a dose and start over. So what about 2 or 3 people out of my first 3 have a dose-limiting toxicity? You may stop, or I might drop down a dose. Fill in 3 more people at that lower dose, make sure it's actually okay, and I might start over. I might say, well, it was flu season. I did have my flu shot, but it was flu season. These people actually they were already immune suppressed, and it seemed like they died of influenza, so we kind of want to restart this. We're not so sure how much of this is really due to our therapy. So you have rules. I don't want you to talk yourself out of your rules. You need to think to have reasonable rules in the first place. But sometimes something weird has happened, and you want to kind of plan these different starts. I will also tell you, sometimes this happens on your first dose. You should always have a backup plan to drop down even from your lowest, supposedly lowest dose. Here it is in a nice chart for those of you who prefer the charts. This is actually taken out of something that Peter Thal at MD Anderson did, which is interesting because Peter Thal really doesn't like this design. But the basic idea is how many patients you have with a DLT and the decision you should make. A few thoughts. So if you have 0 and 3 and 0 and 3, you really had to get here through some de-escalation rule that had to have been applied at a higher dose. Again, sometimes if I had 1 and 3 and then 0 and 3, I might escalate unless it was a de-escalation rule that got me to this 0 and 3. But you have to decide what's your risk of a DLT that you're willing to take. Is it, 16, 17%? Or is it 33%? So what's the risk of having that severe adverse event? It may or may not be that severe depending on how you set your toxicity levels. What percentage of patients are you willing to have that? When you're working with so few patients here, the confidence interval around it is really huge. This is the reason a lot of statisticians now don't use these. They use these different adaptive designs to enroll more people, get more information. But it is important to figure out, if there is nothing available except death, you might be willing to tolerate a lot of toxicity. Or you may say no. So it's important to figure out what percent of toxicity and what toxicities should be your dividing line. So those stars in that previous table, again, you're implicitly targeting a dose with a probability of toxicity less than or equal to 17%. But if you're going to look at the 2 out of 6, then you're saying you're willing to have an MTD with a probability of toxicity about 33%. Key point in these trials, patients do not go onto the next dose level until all the patients at the previous dose level are beyond the time frame where you plan to be looking for toxicity. Do not tell me you need to keep enrolling them, but they're here, they're here. You have to make sure it's safe to put them on the next level. Your patients have to be there, you have to finish analyzing, you have to finish following them, you have to clean their data, analyze their data, and make a decision. Do we escalate? Yes, no. If you do not have the data to decide if you can escalate, you cannot ethically escalate. Again, not a power sample size calculation issue. There are lots and lots and lots of new methods. Almost all of them are better than the method I just told you, but they are also all based on that method. Sometimes in phase 1, now I'm randomizing to multiple different arms. I may have various control arms, might be placebo, might be active control. Why? Because if I'm expecting a lot of toxicity, again, I need to have this comparison to figure out, is this additional toxicity because of my new therapy, or is this my general background rate of toxicity? Especially when I'm dealing with diseases that maybe I don't want to have a lot of toxicity, I want to make sure that they're safer, I might put 6, 15 plus people per arm. Also, if I'm expecting a wide heterogeneity in the effects and the toxicities, I may want to include more people in this phase 1 layer of work. But there are CRM, Bayesian methods, lots of different methods you can use here. Moving into phase 2, this is where we see our screening of new therapies. So phase 2, they've gotten through phase 1. I'm not killing that many people, that's good. Phase 2, I'm trying to still learn a lot about safety, but learn a lot more about my doses. I'm not going to probably look at final efficacy, but I might have some type of biomarker or surrogate outcome that I'm going to determine kind of a pseudo-efficacy that I'm going to power on. Our general idea in phase 2 is it safe enough and is there sufficient activity enough to bother to move this to phase 3, move these to the large pivotal trials? Again, if you look at the old definitions, they'll say, well, should we bother to move it to a randomized trial? These days, almost every phase 2 trial I work on is randomized. It is a rare exception that they're not randomized. Your issues of safety are still going to be really important here. And you have a fairly small number of patients. Might be several hundred, but it still depends. Like, I don't see that many that are more than 100 people per arm, but sometimes it happens. Your problems in phase 2 is that you are more likely to run into the unblinded or single-blinded studies here, have all the problems that we talked about at the beginning of the course. You have issues with placebo effect, investigator bias, regression to the mean. If you're running a single-arm study, you're not really sure what caused those changes to happen. That's the reason that you see this big push. So if you work in industry, if you're one of these pharmaceutical or biotech companies, you're running non-randomized trials, you're running unblinded trials, you tend to move into phase 3 and you get burned. Everything that looked fabulous in phase 2 does not pan out in phase 3. So now I've just wasted sometimes billions of dollars running those phase 3 trials. That's the reason that people are getting a lot tighter in phase 1 and phase 2, because better to stop the product early if it's not producing results. Same thing in academic medicine, folks. You're better to fail small, but be wise about your failures. And with that, then I'm going to talk to you about a non-randomized design, but actually randomized designs have been built out of this. So for phase 2, there's something called a 2-stage optimal design. This was developed out of NCI back, I think, in 1980, if I remember right. And the idea is the premise for a lot of current phase 2 designs. You want to seek to rule out an undesirably low response probability. So let's say only 20% of people respond to treatment. I'm going to rule that out in favor of something that shows useful activity. And let's say I've defined useful activity to be that 40% of my patients are stable or have improved outcomes. So in oncology, they might run a single-arm, 2-stage design using an optimal design and a predefined response definition. It is hard to define a response because then people want to tweak those numbers, right? But again, if we want to rule out a probability of 20% in favor of 40%, let's say I set my type 1 error at 0.1 and my type 2 error at 0.1, right? So I'm willing to make, this is one of those cases where I set them equal to each other. The idea being I'm willing to tolerate a type 1 error. If there's something there, though, I want high power. I really want to see it. So roughly, in this kind of more probability-based setting, I've got a 10% probability of accepting a poor agent and a 10% probability of rejecting a good agent. So if you look in Simon's paper, and if it's not posted, I will ask Daniel to post it up in your class segment. He has this paper with all these different kind of trade-offs of these different probabilities and varying alpha and beta values. This is something that Steinberg and Vinson then built upon in the early 2000s for randomized trials. So we're going to blow this up in a second. This table is table 1 from Simon's paper. We have the difference in the probabilities being 20%. And we've got that we're going to reject a drug if the response rate falls through here. There's something called the optimal design, which is what Simon was trying to promote, and one that it was already out in the literature called the minimax design. So let's look up here. Our null is a response rate of 20%. We want to actually see 40% responders. So what do I do here? What I want to do is initially I am going to enroll 17 patients. So if you looked at the tiny footnotes, we're going to be in kind of this top row here of the bottom row, the bottom chunk. So stage 1, I enroll 17 patients. If 0 to 3 of them have a clinical response, I stop accrual. I assume it's not an effective agent. So what I'm saying is there's no way I'm going to be able to rule out this 20%. So I'm just stopping early. But if 4 to 17 patients have a positive response, I can move on to the second stage. I'm going to accrue up to 37, so I'm going to accrue 20 more patients. And if 10 of the 37 have a positive response, if 11 or more of the 37 have a positive response, then I think it's an active agent. So I'm rejecting if I have 3 or fewer than if I have 10 or fewer. What these other numbers were are, here I have a chance I'm stopping early, right? It might be I stop at 17. If 17 out of 17, I'm done, realistically. But let's look at this. So if I do the standard sample size, under the null hypothesis, I have to enroll 26 patients. Probability I'm going to end under the null is 55%. It's pretty good. So here it is in words for you what I just said. Under the design, if the null hypothesis is true, there's a 55% probability of early termination. This is one reason we like phased designs, right? We get out early. It's not 100% sure, but 55% is better than 0%. On average, in that 2-stage optimal design, under the null, I enroll 26 patients. If I used a 1-sample test of proportions, I need 34 patients. So 34 is fewer than 37, but I don't have that chance to stop early. And that's what I'm gaining in this 2-stage design. If I use a 2-sample randomized test of proportions, I have 86 patients per group. And that's the reason you have this lure of a single-arm study. Why do I want to have 2 arms and randomized when I can get some information? Maybe it's a problem, but I have so many fewer patients. It takes me less time and less money. This is what you have to balance as an investigator. Again, there are a lot of newer methods available. They all end up citing Simon, so that's why Simon is here. So let's talk about historical controls now. In historical controls, let's say I've got a rare disease. I want to double their survival. Survival right now is 15.7 months, and I want to, with my new intervention, double that median survival to 31 months. I say I'm going to have a type 1 error of 0.05. I want a one-tailed test, and I want my power to be 80%. So why one-tailed? Basically, if they're dying, then this is going nowhere, right? So if my survival is less, no, that's it. So and also the reason that they went ahead and did a one-tailed test is they said, this is a rare disease, and we have to do everything we can to try to have a reasonable test. And because they're like, it's going nowhere, and they swore they were going to publish it either way, they're like, yeah, it's interesting, and it will be clear that this is wrong. So they had a one-tailed test. You need 60 patients for this, and we'll talk about how you figure this out. About 30 in each of the 2 study arms. They come back and they say, okay, Dr. Johnson, I can accrue 1 per month. It was actually a different doctor they were talking to, but he let me share the example. So if you can get 60 patients, 1 per month, that's 5 years. To just enroll them in your study, let alone follow them in the study. They needed 36 months of follow-up, so that's an additional 3 years. So now it's going to take me, in best case scenario, 8 years from the time I start enrolling patients to when I stop collecting data. Yeah, that's a long time to wait for an answer. Plus, realize it probably took you a couple of years to get this study up and running. It's going to take you a little bit of time to analyze and publish your data. So they said, well, we want to use historical controls. We have a bunch of patients. They were controls in other studies. We want to use their data and put all patients that we have on the new therapy and compare them to the old patients. So there was this old data set. We had 35 patients. They'd been treated in National Cancer Institute with this disease. Problem is they were initially treated from 1980 to 1999. Of those 35 patients, at the time they were looking at the records, 3 of them were still alive. Median survival time for the historical patients, so where they'd gotten the value from, was 15.7 months. And this is basically kind of like this observational study, right? But, 1980, it's 2015 now. There's a lot different. When we diagnose them, how we diagnose them, what measurements you're taking, how you're recording it, how you decide that people are progressing, the treatments that we're giving them, not just to treat their disease, but everything else, to help their nausea, to help their anything. It's all different. So you have to decide, do you use this data? Is it really controlling for everything you need to control for? Does anybody else have data? Can you work with people around the world to do this protocol? How do you do it? So these are the problems that come up in phase 2 studies. You've got to constantly be weighing the advantages and disadvantages of your study designs. You may have a single-arm study that has small sample size. That's good in some ways, but you've got no control information. You may have a 2-stage design. Again, you might stop early with a small sample size, but if it's a single-arm 2-stage design, you've got no control. You might have the correct responder, non-responder rules, or you may not. Are you sure that that difference is really what you care about? Can you make a binary distinction to really even define a responder? Historical controls. Again, it's great because you can use a small sample size, but you don't know how accurate that control group really is. You might decide to run multiple arms, sometimes randomized, sometimes not. Sometimes I see people run like 8+ arm studies, but then they have no control arm. They're just comparing all the different doses to each other. I'm like, that's great, but what if none of your doses work or all your doses work the same? Like, you don't have a way to compare. So if you're going to be running multiple arms, please, please, please have something with some control in it, because it could be that every dose does something, but it all does the same thing. The downside is it takes a lot more people. That said, I see more and more phase 1 and phase 2 studies enrolling hundreds of people because they want to make sure what they're moving forward into these final large pivotal trials is what they've got the best shot at, and that they better understand a lot of the safety information, the pharmacokinetics and pharmacodynamics, and a lot of other information about potential efficacy in trials, especially in multiple different populations, because that's where we do our trials. So we're going to go a little bit through this phase 3 example. And here, this is actually a survival example. So our primary objective here was to determine if patients with metastatic melanoma who underwent this procedure had a different overall survival compared with patients receiving standard of care. So remember, we have to then define standard of care. But they were trying to do all this, and the idea was that we were going to have a 2-arm randomized phase 3 single institution trial. I don't see many phase 3 trials done at one site, but this one was going to be. So they came to us and said, how many patients do we need to enroll? We want to have a 1-to-1 ratio between the arms, so we're going to equally randomize to the 2 study arms. We want 80% power to detect a difference between 8-month median survival and 16-month median survival. Again, then you need to look and say, okay, what's currently on the market? What's going on? Do these values still make sense? They had a 2-tailed 0.05 test they wanted to run, and they wanted to do 24 months of follow-up after the last patient had been enrolled. And they were planning on doing 36 months of accrual. These lovely things are called nomograms. So I have plenty of books with lots of formulas, and I have to say, actually, of all sample size calculations, probably like survival analysis ones the most because you get to move a lot of different things. But some of the best statisticians I work with keep photocopying this picture. There's this paper that basically has different figures and the number of patients per treatment group. So they have different pictures for alpha and beta combinations. The dashed lines are for 2-sided tests and the solid lines are for 1-sided tests. So what do? I said, okay, first, my follow-up period is 2 years. So you draw a line that is perpendicular to this down to intersect the line for your accrual period, which was 36 months. So you draw that line out. And then where line 1 and line 2 intersect, you draw straight up, parallel to this y-axis with the number of patients. You draw straight up. You say, all right, R is the ratio between the 2 median survival values. They said that they wanted to basically double median survival from 8 to 16 months. They had a 2-sided test. So we go from the dashed line for R equals 2.0. We draw a line parallel to this x-axis. Boom, 37 patients. So they have all these pictures, and no joke, you'll see the statisticians like drawing with their lines. You can do it online now. They have a little app. Schoenfeld and Richter made these pictures back before we had computers that easily calculated this for us. Now most statisticians will honestly run a bunch of simulations and simulate your sample size for you, taking into account a lot of different issues about your study design. But it's a nice check, I think, for folks. It's a low-tech check that you can use. You can use formulas, you can use simulations. Almost everybody can draw straight lines, as long as you have a ruler or something to draw them against. Oh, I'm sorry, it wasn't 37, it was 38 per arm that we needed, so the decision was to try to recruit 42 people per arm for a total of 84 studies because they assumed that they were going to have censoring, people were going to drop out, et cetera. They said that they anticipated approximately 30 patients a year entering the trial. We said, yay, that works. Because sometimes they come back and you're like, oh, you had an accrual period of 36 months. Well, maybe that's not going to work anymore because of the number that you think you can get per year. We're going to have to jigger and do this estimation back and forth. So 30 patients a year, you think in 3 years I'll get 90 patients. I should hopefully clear my 84 that I need. So again, nomograms. I show this picture because you're not always, sometimes you're up here, you're not always down here. So let's think of non-survival simple sample size. Let's start off with a 1-arm or 1-sample study. Then we're going to move into a 2-arm sample or 2-arm study. Cheat trick. If you have 3+ arms, calculate the per arm sample size for a 2-arm study. Use that per arm. Doesn't always work, but it's typically a reasonable estimate of how many people you need per study arm. So let's go back to a single sample test. I've got a new sleep aid. I want to look at baseline sleep time after taking the medication for a week. I'm going to run a 2-sided test, alpha equals 0.05, power 90%. And the difference is on average 1. So I think on average people are going to have 4 hours of sleep at baseline, and I'm going to move them to 5 hours of sleep after my intervention for 1 week. Standard deviation, 2 hours. These are not real numbers. They are numbers to make this thing move. So now I've got my equations. I've got this one sample test. You'll notice that there's no 4 in front of this. That's because the 2 and the 2 before were because we had a 2 sample test. So 2 times 2 equals 4, which is why that 4 is there usually. But I have kind of that critical value for my type 1 error. It's a 2-sided test, so it's divided by 2. Critical value for the power, variance, and the difference of interest. So for an alpha equals 0.05 2-sided test, my z-value is 1.960. For the 80% power, my z-value is 1.282. Add those together and square it. My standard deviation was 2, so I'm going to square that. So I have 4 for my variance. My difference of interest is 1 squared is 1. If I calculate this, it says I need 42.04 people in my sleep study, which means I need at least 43. Let's say now I want a 2-hour difference in sleep. So I want to measure them go from 4 to 6, not 4 to 5. My sample size shoots down to 11. How did I get 11? Well, remember what I said. It's easier to see big differences. It's easy to see if people have, on average, 2 hours more sleep. It's easier to see that than 1 hour. It might be harder in reality to make that change happen, however. But I took this value in the denominator. Remember, it's a value squared on top of that. So you can, if you're more math algebra oriented, remember the formula. If you're not, just remember, if you increase the difference of interest, you decrease your sample size for the same power. But a sample size of 11 leads us back to one of those little tricks in the front. I found this using the z critical values, but it's a small number, so I need to do this iteration with the t's. If you are not a mathy person, do not stress. Your computer will typically do it for you. If you are trying to plug numbers in, you need to stress and remember this. So you first calculate it using a regular formula. Then you take that sample size. You're going to have n minus 1 degrees of freedom down here for a simple 1 sample test. You're going to find the critical value of t for 10 degrees of freedom in this case. Recalculate sample size using the t's instead of the z's. You're going to find a sample size of 13. You repeat this whole thing. You iterate it down till you get a single sample size that sticks. Sometimes you have to iterate this a lot. Again, only for those that bother to do this essentially by hand or want to program their own do you care about this. Although you should check, this auto formula, is it using the t or is it using the z? If it uses t's, you're good. If it uses z for the critical values, you may want to use a different formula. So let's say my power. Before I wanted a power of 90%, now I want 80%. Oops, I'm sorry, I had the wrong z there before. My sample size goes from 11 to 8. Again, I need to be thinking t distribution, not z distribution. But this value in the numerator just got smaller because my power got smaller. If your power you're interested in is lowered, your sample size that's necessary is lowered. Standard deviation. Oh, and I should say here, you do not need an actual sample size of 8. You need something larger because your math for all your regressions are going to fail. But this is kind of your starting point, is the 8. Standard deviation goes from 2 to 3. My sample size has now moved from 8 to 8 to 18. Why? Because I square that 3. It's in my numerator. Increased variance means you need an increased sample size. let's say you remembered that Dr. Johnson told you to do a randomized 2-arm design. Don't do these single-arm studies. You can if you need to, but you're like, I'm going to do better. So you take your original design. Remember, we needed 43 people in the 1-sample design if we stuck with our original numbers. 2-sample study needs twice that in each group in order to account for the variance. Because now you have variance in 2 different study arms. You have even more variance. So because of that, I need 85 people in each study arm, or 170 people total. And then you go, well, didn't Dr. Johnson say sometimes single-arm studies were okay? So again, you're going back and forth on this. Here are your 2s. Those 2s are because you have a 2-sample study. Again, 5-arm study, calculate that sample size per study arm, multiply it by 5. It doesn't consider all the multiple comparisons. It depends on the analysis techniques you're using, but this is your rough estimate to get you started. So think about it again. You change the difference of interest from 1 hour to 2 hours. That 170-person study goes to 44 people total. Sounds pretty sweet. Changing my power from 90% to 80%. The sample size goes down to some more. My standard deviation maybe needed to be a little bit higher. My sample size has gone back up. So in conclusion, a lot of things drive your sample size. You can either remember the directions or you can remember the formula, and that will help you figure out the directions. But just remember, basically everything about your study is driving that sample size. So are there other designs? Sure, there are tons of designs. If you do a matched pair design, essentially this looks like the 1-sample formula. If you're doing means, you're going to do the formula for a paired t-test. It's going to look at the mean difference from the paired data and the variance of the differences. And proportions. Proportions are actually based in discordant pairs for your sample size calculation. The textbook has various examples with paired designs, 2 and 1-sample means, various proportions, and we also talk a little bit about how you take pilot data and design your next study. I mentioned I don't like Cohen's effect sizes, but it is very popular to use them in sample size calculations when you don't have any other information. And Cohen himself, who has a very commonly used textbook in the psychological literature, says don't use them unless you don't have a choice. A large effect size is typically 0.8, medium is 0.5, small is 0.2. Medium, again, yields you the same sample size regardless of what you're measuring. That's the reason Cohen says, please don't do this. So what are some changes? If you have unequal numbers in each group, the ratio of cases to controls, you want to use this if you have, say, a certain lambda set of patients randomized to the treatment arm for every patient randomized to the placebo arm. You can also use this in a case control setting. So the idea here is that you're going to have different variances. You're going to kind of use this ratio, calculate the sample size for one group, and then use that to calculate sample size for the other group. The shortcut way of doing this, let's say I'm going to use equal variance sample size formula, so I assume both groups have the same variance. I want to look at the total sample size, calculate that, then increase it by this factor k. So let's say I'm doing 2-to-1 randomization. I calculate the total sample size. If I had 2 equally sized study arms, it's 26, so I was going to have 13 per arm. I say, okay, 26 times 2 plus 1, because that's my ratio, squared, divided by 4 times the 2. And I find out that I need 30 people in my study instead. So I would have 20 in one group, 10 in the other. Why might I do this? Well, sometimes I have a device. Let's say I have a new artificial knee, and I want to know how many knees I need to make. and I want to do an unequal randomization. This might be a case where I use that. Sometimes I have a fixed number of cases. I've already made the knees. I only have so many of them to use. So if my sample size calculation said I needed 13 per arm, but I only have 11 of the knees, I want the same precision, then I'm going to calculate how many people will be in my control arm. I back calculate that using this formula. When you're doing survival analysis and other types of analyses, you may have a cohort of, say, those exposed and unexposed people that we talked about. You have this relative risk that you're interested in. I know the prevalence in the unexposed population. I need to figure out the number of events. So the risk of an event in the exposed group divided by the risk of the event in the unexposed group, and back calculate the number of events I think I'm going to be seeing. So I have the number of events in my unexposed group, calculate the number of events in the exposed group. Then calculate how many people I need to see the events. Remember stability issues. In logistic regression, there's this big discussion, and a whole bunch of simulations were done, that you need at least 10 subjects for every variable investigated. When you look at principal components analysis and a lot of other methods that get used, you see everything from 10 to 100. It's all sorts of crazy. Some people don't say you have to use balanced designs. Other people say you have to use them. I'm just going to say, if you have balanced designs, you have the same number of people in each study arm, it's easier to figure out your power and your sample size. It is easier to handle, but, I have a job to work on complicated designs. So you can do simulations. You can do pretty much anything you want as long as you can do the work to make sure it's going to be rigorous and valid. So what about multiple comparisons? If you have 4 groups and you want to do all 2-way comparisons of means, you have 6 different tests. But sometimes you say, well, I have 4 groups. I have 1 placebo arm and 3 different doses, and I actually just want to compare each dose to the placebo. I'm not going to compare the doses to each other. So you have to figure out exactly how many tests you really are going to run. The simple way people do it is this Bonferroni test, right? They divide their alpha by the number of tests. It's common, there's a long literature, but it's super conservative. And in some of your high-throughput laboratory tests, it can be worked out. Ed Korn, I think it was, worked this out, and maybe Lisa or somebody else in that branch. It said, there were cases that it was physically impossible for them to have any statistically significant results after they accounted for multiple comparisons. That's not an experiment worth running. This comes up a lot in these microarray, proteomics, other omics types of experiments. Many times people say, just set your alpha and your beta stricter. I tend to agree with this. Just set a really strict type 1 error level that you're going to adhere to and test everything against that. If that's what you want to think about for false positives, false negatives, there's also family-wise error rates and all these other things, but I think just set strict up front and plan for that. So what are some of these rejected statements? Again, this St. George's Hospital, I'm looking at this link. I think this is the correct link, but I don't remember if I double-checked it this morning because they keep moving some stuff around. Anyway, you can put the name into your favorite search engine and find it. St. George's Hospital Medical School has one of the best statistics guides for research grant applicants I've ever seen. Many times I was can I just copy and reference it and be done? I think it's very applicable across any country that you go to. So one of my favorite parts, because I see it way too often, is people who go, me too, me too. It's no. You must justify your sample size in every single application. So this is an example, an exact quote. A previous study in this area recruited 150 subjects and found highly significant results, p-value equals 0.014, and therefore similar sample size should be sufficient here. Well, they could have been lucky Could have been random sampling variation, could have been a lot of other things. You need to give an estimate of what you're expecting for the variance, for the difference, what your type 1 error is going to be, etc. You have to give all those little elements. It's good to know that some study who had similar elements found something significant, but honestly, if a lot of studies have already found this, why are you doing your study? No prior information. Now, this is always trouble because sometimes you do have no prior information, and what you're trying to do is run the little baby first pilot. But the quote in the application, for a substantial sample size application, I should say, sample sizes aren't provided because there's no prior information on which to base them. All right, we have to know how many people, how many animals, whatever, you are enrolling in this study. Find something that's published previously and try to extrapolate from that. Conduct a small pre-study, but if this is your application to conduct the small pre-study, then you probably, in fact, don't need sample size calculations. Don't tell me the sample sizes aren't provided. Tell me, like, this is where you are and that you're going to use this amount of information to then calculate appropriate sample sizes. But this is what you're gathering in order to do it. But if I can easily find a bunch of information that could have informed your sample sizes, I'm probably not going to have a very happy review for you. Variance. No prior information on standard deviations. Well, again, this might be where you talk about detecting the size of difference in terms of the standard deviations. This is where those effect sizes that Cohen and others, and their different effect size values for different regression models, but this is where that can be useful. Again, it's your starting step, but it's not going to be what you use for a pivotal or final study. Now, the number of available patients, Wendy Weber and others are going to talk a little bit more about this, but you have to make sure you have patients to be in the study. A few things. You will not get all patients to be in your study, number one. In fact, you're lucky if like 5% agree to be in your study in most cases. Number two, feasible is important. It does not tell me about your power. So this is, again, a direct quote that they got at St. George's. The clinic sees around 50 patients a year, of whom 10% may refuse to take part in the study. All right, I don't think that's realistic, that only 10% will refuse, but we keep moving on. Therefore, over the 2 years of the study, the sample size will be 90 patients. Let's say a miracle occurs and they get their 90 patients. I have no idea what the power is for this study. I know nothing about the differences that they want to detect. I don't even know what they want to measure. I don't know about the variance. I know nothing. It's okay to give this information and then say, and with 90 patients, our power will be X based on the following assumptions. That would have been okay, but that sentence never appeared in the application. So you do have to balance feasibility and power, but sample size isn't decided just on available patient numbers. So what are some of our resources and conclusions? What impacts your sample size? The differences, the standard deviations or the variance, your type 1 error that you're willing to tolerate, and your type 2 error that you're willing to tolerate. Number of arms or samples, 1 or 2-sided test. Are you randomizing? If you are, what type of randomization are you using? What are the analysis plans? If you don't have an estimate of the variance, make sample size or power tables, make graphs. You should, in fact, always, even when you think you have good estimates, they may not be that good, you're always going to want to make tables and graphs. Show a wide variety of possible standard deviations, of possible differences, and protect yourself with a high sample size if it's at all possible. So when I was reviewing grants for the NIH, the top 10 statistics questions I had to ask back to investigators. The exact mechanism they were going to use to randomize their patients. Don't tell me you're just going to randomize them. Dr. Joaquin went through a whole bunch of different ways you can randomize people. Tell me more information. Tell me when, etc. They would say they're going to stratify. I'd be like, why are you going to stratify? What are you going to stratify on? The EMAs, the European Medicines Agency, actually provides different information on something called dynamic allocation. So if you're going to use different types of allocation schemes, they may or may not be acceptable to different regulatory bodies worldwide. Or they may be acceptable in pediatrics, but not acceptable in adults, acceptable in oncology, not acceptable in cardiovascular disease. Blinded or masked personnel. Who is doing your endpoint assessment? Who is masked? To what are they masked? Are you compromising your blinded study with something else that you were doing? Many times I had to ask people to list every single hypothesis, the hypothesis test, the specific analysis, and the specific sample size or power justification for those hypothesis tests. That should be very clear in your grant application. Somebody shouldn't have to ask you for this. It should also be clear in the statistical analysis plans that go with it. And it needs to match everything else in your protocols. I would need to ask how or if they were adjusting for multiple comparisons. Some fields get very, very picky about multiple comparisons. Others say, well, if you're honest and you report everything, you can say I'm not adjusting for multiple comparisons. Typically, most of your regulators in pivotal trials will make you do what's called a statistical hierarchy or some way to control the type 1 error. So it may or may not be technically adjusting for multiple comparisons, but you are kind of doing a conservation, they call it, of the type 1 error. I would ask if they're testing for effect modification. Are you doing any interim analyses? And if so, what exactly are you analyzing? When? What's the error spending model and what are the stopping rules? So those error spending models are, again, to conserve your type 1 error. And I would ask if this was accounted for in the sample size calculations. Why? All those ways that you do interim analyses impact your sample size calculation. What is your expected dropout? They would tell me about their sample size, but not the expected dropout, and did they plus up in order to account for this? How do you handle the dropouts and missing data in your analyses? Again, those methods may also impact your sample size and your power. And then I always had to ask about repeated measures and longitudinal data, almost always. Are you using a linear mixed model? Are you trying to use repeated measures ANOVA? Repeated measures ANOVA is an awful method. It's very antiquated. It has a lot of assumptions. The newer methods have a lot fewer assumptions. They're a lot more robust. Maybe they're going to use generalized estimating equations. But again, what is your actual method? How are you collecting the data? What's the study design? What are the analyses in order to actually test that data? All of that goes into figuring out your sample size and the analyses and conclusions that you can draw. So Paul actually has a version of the sample size lecture online. So when we were in Brazil teaching, they filmed us and put everything on YouTube. So if you want, you can watch Paul give a similar type of lecture back about a year ago, right? This might be more depending on when you're watching this lecture, so September of 2014. He focused more on larger trials and on those kind of later phase studies, but he has some really great examples. So I would encourage you to actually look at his video. I want to say it was maybe 90 minutes. It's under 2 hours for sure, and you can listen to his questions. But speaking of that, if you have any questions here, we'll be on the discussion board for the next 2 weeks, and go ahead and at least 2 weeks from when the lecture is scheduled. I can't promise you 2 weeks from when anybody outside of this room is watching it, but ask your questions on there and I'll try to answer them. If you want, the last several slides here are basically just general books and other resources, sample size calculations that may be useful. I'll talk to Daniel about posting at least his first article because it might be hard to find. The rest of the articles should be fairly easy to find. But if you have trouble and want them, let me know and we can post them to the class website. There's also a lot of regulatory guidance that talks all about sample size issues. So you may want to look through those. Thanks, and have a good evening.